So thank you very much for that kind introduction, and thanks to the organizers for the invitation to speak here. And so I'm going to talk about complex photophysics of fluorescent proteins. And because in a 1 hour lecture, you can actually only learn about 3 things, you've seen many of these concepts in probably all of the other lectures that you've had, but I was assured that repetition was a really good thing, particularly for students who are new to a topic. And because you only learn 3 things from a lecture, hopefully you'll get something new that you didn't get in some of the other lectures. So I'm actually going to start by recapping some of the terminology that we've come across in some of the talks that have been presented so far. So people have talked about photobleaching, which is the irreversible destruction of a chromophore, and I'm going to talk a little bit more about that in my talk, how we measure photobleaching, what the implications are for imaging, and how we might go about trying to engineer more photostable probes. We have heard a lot in passing about photochromism, that is both reversible and irreversible. Properties such as photoactivation, where you can, upon illumination, turn on fluorescence. Photoconversion, which is generally thought of as irreversible, upon illumination changing the color of the fluorescent probe. Photoswitching, which as the name implies is a reversible process, a light-induced transition between, for example, a light and a bright and a dark state. And then we've also heard blinking dark state conversion, sometimes also referred to as reversible photobleaching. All 3 of these generally are the same thing, although there might be many different mechanisms of blinking. And this is a spontaneous process really in all fluorophores, certainly in all fluorescent proteins. We also have seen examples of different kinds of photochemistry where we refer to a process as chemistry often if it is involving a change in the electronic structure or in the chemical structure. So there are examples of cis to trans isomerization of chromophores, particularly the fluorescent protein chromophores. There are many examples of making and breaking covalent bonds, such as decarboxylation or cleavage of protein backbones, excited state proton transfer, and then of course redox processes that can happen in the excited state. And so I'm going to try to touch on all of these. So if you remember many of them, try to pick a new one to try to remember from this talk. Okay, so I also wanted to highlight a number of reviews. There are many in the fluorescent protein field. So what I tried to do was pick some of my favorites that I think highlight some of the things that I'm going to talk about. So in particular, if you are interested in the photophysics and photochemistry of chromophores in fluorescent proteins, I think all 3 of these are really excellent reviews. This one by Anna Krylov and colleagues, this one actually by our organizer Dominique, and by 2 other attendees and speakers, Karen and Ulrich. This one here are really wonderful reviews on photochemistry. I include this as a reference because I use a number of their pictures of fluorophores, and I wanted to acknowledge that. So there's some pretty pictures of chromophores. And then both of these 2 reviews are more practical summaries of fluorescent proteins, where this guide here still has probably one of the best discussions of how we measure photobleaching, and that is then followed up on this really nice overview of quantitative assessment of fluorescent proteins out of David Piston's lab, where he also discusses photobleaching as well as oligomerization, which I'm not really going to talk about. Okay, so I'm going to remind you of where we started this course. So I think Fabienne presented the equivalent of these photos of a photocycle on the very first night. So this is one example that we start our process with absorption. We hope that our fluorophore will then release give off a photon in order to go back to the ground state, and that is the process of fluorescence. But unfortunately, there are many different pathways to get back to the ground state, and we generally refer to those as non-radiative relaxation because we're not giving off a photon. When we're in the dark state, however, you've heard of the process of blinking. So it is possible, instead of going back down to the ground state, that you can convert to a dark state. Dark state really just meaning that it's not bright, you can't see it, it doesn't give off a photon, or so dim that you can't see it. So you can convert to a dark state, and we generally refer to that, or I'll refer to it primarily as dark state conversion. From the dark state, of course, you can relax back down to a ground state, which in this case may be different, is probably different from your original ground state. However, a dark state itself, if you are illuminating a sample, even if some of the molecules are in the dark state, they can absorb photons, so you can get absorption within dark states. Of course, molecules in the excited state can bleach, so this would be different than a non-radiative relaxation, where this process would imply that you could reabsorb another photon and go around the cycle, but bleaching really just takes you off pathway because it's a destruction of the chromophore, and then you can no longer go through the photocycle. You can bleach both from an excited state or from a dark state. Dark states can be photoreactive, and so that's something to keep in mind. And then I'm not showing, of course, all of the conversions that can occur. These dark states could be triplet states or could exist coincidentally with other states like triplet states. And of course, in all cases, if you use really intense illumination, you can get higher order transitions from the excited state. Okay. Oh, sorry. Lastly, if you go through a pathway like this, then there's some rate at which the molecule will relax back down to the ground state, and can then go through a photocycle again. Okay, so Anna Krylov in this review talks about many of these processes. I've just rearranged it here, or she's rearranged it in this figure, showing that if this is the ground state chromophore, when you absorb energy, you promote an electron to an excited state. And then she shows some of the different processes that can occur. That excited state chromophore can fluoresce. We often use the radiative rate, or K sub R, to discuss this process, the efficiency of giving off a photon. The processes that generally are grouped together as non-radiative relaxation, we often have this terminology here, KNR. Of course, there can be photochemistry, there can be chemical transformations, or there can be redox changes, and then you can flip this spin that would give rise to a triplet state. Okay. All right, so what are some of the photochemical reactions that can occur in the excited state? One of the most common ones that we observe is a cis-trans isomerization. So this is shown the chromophore of GFP, and although I'm talking about fluorescent proteins here, obviously many of these same principles are found in small molecule fluorophores as well. So shown here, you can see, I highlighted it for you non-chemists, around this double bond here, there's a different conformation here. So this is the cis configuration and the trans configuration. And it's not hard to imagine that if you undergo this rotation in the excited state, that this phenol moiety here is going to be in a different chemical environment. And indeed, this is we've seen a lot of examples references to Iris FP, and this is some beautiful work by many of our speakers and organizers here, where they looked at the crystal structure in different states. And indeed, you can see this is a case where you have the molecule in this configuration, and you see now the chromophore is deprotonated and interacting with this serine 142. But when it undergoes cis-trans isomerization, it's in a different chemical, and ooh, I pressed the wrong button. Sorry. When it's in a different chemical environment, and now this is protonated and interacting with this glutamate 144. So a different chemical environment. Okay, so in addition, we have seen examples of excited state proton transfer. As Peter very nicely showed in his talk, in the GFP chromophore environment, there is this beautiful proton relay. So you can see many hydrogen bond donors and acceptors. And what can happen here is that in the excited state, you get a rearrangement of these hydrogen bonds that leads to excited state proton transfer. So what had been a protonated chromophore is now deprotonated, and that of course shifts the excitation from 405 to 488 in the case of GFP. You can also break covalent bonds, and this image from Dominique's work is, I think, one of the more beautiful images of showing this is two crystals in two different states. You can look at the X-ray, the electron density here, and what you can see is that in this case, this is the chromophore here shown in green, but upon excitation with 405 nanometer light, you break this bond. That leads to extension of the conjugation. So extending the conjugation shifts the wavelength to the red, and that is an example of breaking covalent bonds in the excited state. Okay, so these are some of the many photochemistries that can occur. So the first example that was identified is that of the photoactivatable GFP, which we've seen used in many things like palm and other kinds of microscopies. So this was discovered by George Patterson and Jennifer Lippincott-Schwartz in 2002. And this is an example of light-induced breaking of a covalent bond. What they had noticed is if you take wild-type GFP and you look at the absorption spectrum here, this is the native absorption spectrum in these closed circles. But when the protein is photoactivated, you shift from this 405 peak to this 488 peak. So they thought this was very interesting, but they wanted to be able to tune it better. So you'll notice that in the native state, there's still a 488 peak here. So the contrast, the difference in signal at the 488 wavelength is not that great in the wild type. So they actually found a single mutation, this T203H, enabled them to decrease this in the native state such that upon photoactivation, only upon photoactivation did you see this peak at 488. There was also a slight redshift in the emission. And it was discovered that this was due to decarboxylation of this glutamate 222, shown here. So upon 405 nanometer excitation, you lose carbon dioxide and a proton, and you generate what we think of as the GFP chromophore that absorbs 488. And a similar process also exists for photoactivatable M-cherry. It's really the same chemical reaction here where you're losing carbon dioxide, although there's also some oxidation. Okay, so that's an example of photoactivatable FPs. How do we use these outside of palm? So we've heard a lot of examples of how we use photoactivation for palm microscopy, but actually these were really useful as biological tools even before the advent of PALM. So they can be used for protein tracking. For example, measuring the rate, direction, diffusion constant, the mobile and immobile fractions, rate of turnover. So the idea is you can just illuminate a certain spot and then track those proteins over time. You can do this with individual organelles. So if you were looking at vesicles within a cell, you could illuminate and photoactivate a single vesicle and then watch its movement. And then, of course, you could use it for cell tracking as well. You could turn on just these cells and, for example, in development, watch where they migrate to in a developing organism. And some of these applications are highlighted in this review. Okay, so this is just one. I wanted to give you a real biological example. This is a paper out of Richard Yule's lab from the NIH. So they were studying this bacterial toxin called diphtheria toxin. It gets into mammalian cells via endocytosis, and then it gets cleaved. It's a one protein, but it gets cleaved into many bits inside of the endosome, and then a piece gets into the cytosol. And what this does is it inhibits protein translation and then leads to cell death. So they were trying to understand the mechanism of how this diphtheria toxin traffics in a cell, and in particular how it gets released from the endosome. So what they were doing is they were studying endosomes, and as a marker for endosomes, they were using this protein Rab5. Rab5 often coats endosomes and marks certain populations of endosomes. So they tagged Rab5 with the photoactivatable GFP. And what they found, you can probably tell by comparing, this is wild type cells and these are cells treated with this diphtheria toxin, that when cells are treated with this diphtheria toxin, the endosomes get really big. Okay, so that's one thing. Here they're small, here they're very big. So this toxin increases the size of vesicle fusion, which they said was due to the size of endosomes, which they said was due to increased recruitment of increased fusion activities. But then what they did, how they used this photoactivation feature, is that they photoactivated individual endosomes, and then they watched the rate of fluorescence decay of the Rab5 on the Rab5. So what they were trying to access was, what is the lifetime of Rab5 on an endosome? How quickly does it exchange off the endosomes? And what you can see here is in red are control, and in blue and green are two examples of this diphtheria toxin. And so they use this photoactivation to show that you get slower exchange of Rab5 from the endosomes. So what these two features suggested is that what this toxin is doing is it's increasing the size, it's making these endosomes much bigger, and it's preventing maturation. So these guys stay as early endosomes. They don't completely mature down the pathway, and they suggest that this is important for releasing this part of the toxin. So that's one example of how photoactivation can be very useful. Okay, I'm actually not going to cover photoconversion as much because Ulrich did it so nicely yesterday, and I don't have as pretty movies as he has, but suffice it to say that there are a number of photoconvertible proteins that similarly have irreversible light-induced chemical transformations. So we can have decarboxylation, we can have oxidation, and we can have backbone cleavage, and these are just some examples of blue to green, orange to reddish, and green ultimately to red. And how these can be used really nicely is for cell tracking. The same way that you can use photoactivation, but the problem with photoactivation is before there's light, you have nothing to look at. So that's really challenging. Photoconversion can be really powerful because you can see your cells, for example, in green, and then you can choose which cell you want to turn on or to turn red. So that is how it's used here, where say you want to follow a pathogen as it infects an entire dish of cells. You could have your pathogen expressing a photoconvertible protein, for example, green, and then you could mark certain pathogens in red, and then you could watch what happens to those pathogens as they maybe transfer from one cell to another. You could do this in a mouse, and this has been done. Ulrich actually showed some very nice examples of organismal development. But if you have a population of cells, you could photoconvert them and then track the birth dating, so track which cells live longer and which cells are newly synthesized, because the new ones will be green and the older ones would be yellow because they'll have both green and red. You could just convert an individual cell and track differentiation, or convert an individual cell and then look at migration. So if you're interested in cancer metastasis, for example, and you want to look at migration of cells from a site of origin, this could be really powerful. If you're a cell biologist, you could actually use this for tracking organelles inside of cells instead of tracking cells in an organism. So this has been used, photoconvertible FPs have been used to look at mitochondrial fusion events. So when mitochondria come together and they mix contents, it's been used to monitor maturation within the Golgi and the ER, to track protrusions of cells. So there are a lot of examples of how you can use this. And I just pretty much wanted to show you this picture because I thought it was really striking. So this is an early example of using Caeta in making a transgenic mouse. And I just thought this was incredible that you could see this photoconversion on the entire mouse by illuminating. Here they were illuminating the animal for different amounts of time, and you see the conversion. In this study, they actually used it to track cells. They photoconverted cells in a lymph node and then tracked them over time. So what is the residence time of cells within a lymph node, and where do they track in the body? Okay. So reversible photoswitching proteins, again, we have heard a lot about these. I think Drampa, I'm pretty sure Drampa was the first example of this, converting with 405, converting to the bright state, and then 488, converting to the dark state. There are many other examples, so this can happen with red fluorescent proteins too. And I think where these became very popularized is in the use of using these for resolved microscopy, which you have heard some about over the course of the week. And recently, I think it's true that probably the best fluorescent protein for resolved is now this reversible switching GFP2, which was developed by Stefan Jacobs. And in this case, he's showing, this is an absorption spectrum, and this is showing the switching of Drampa and the switching of RS-GFP2. So essentially what they tried to do is they tried to engineer a protein that would switch much faster, and you can see that in this comparison. And then further, this was the RSGFP, and they further engineered it to be faster a little bit later in this eLife paper of taking the RSGFP and making RSGFP2 where it switches even faster. And now they've used this fluorescent protein with resolved in animals such as Drosophila. So trying to be able to do super resolution in animals. Okay. So I wanted to turn now to photobleaching because we have mentioned it a lot, but we haven't talked about it in a lot of depth. So photobleaching, to remind you, is a general term to describe a variety of processes that lead to destruction of the fluorophore. So what causes photobleaching in fluorescent proteins? The mechanisms aren't actually that well understood, but there is evidence that there's dependence on oxygen, and there's evidence of electron transfer. So one example of evidence of dependence on oxygen is from this paper here from a number of years ago, where Nathan Shaner was trying to improve the photostability of two fluorescent proteins. And what he found was that if you remove oxygen, so you simply deoxygenate the media and measure the fluorescence decay, you can slow down the fluorescence decay. So an example of that is here is a photobleaching curve in this solid line, and when you take this guy, if you look at this, one of these dashed lines, basically if you remove oxygen, you shift it here, so it decays more slowly. And these are two other examples where here you have fast decay, you remove oxygen and you slow the decay. You remove oxygen of this guy, you slow the decay. However, if you're looking carefully, if you're not asleep yet and you're looking carefully, what you'll notice is that they engineered this protein mOrange2, and here removal of oxygen really had no effect whatsoever on the fluorescence decay of that particular protein. So there's evidence that it is dependent on oxygen, but that's clearly not the only decay mechanism. Okay. There are many possible pathways. This is a nice paper by Anna Krylov's lab where they show that there is also evidence of photo reduction in the excited state. So just to remind you, if you have your chromophore, you absorb light, you're in the excited state. You can convert to a triplet state. But from this state, you could either get reduced or you could get reduced or you could get oxidized, but you could also react with oxygen via this pathway. If you convert to the triplet, you may know, how many people have taken introductory chemistry? Everybody? Okay, so you may know that oxygen is a triplet, has a triplet ground state, and so if this converts to the triplet, then that increases the probability that you can get oxidation, a reaction with oxygen to produce singlet oxygen. And similarly, the presence of singlet oxygen has been observed upon photobleaching of fluorescent proteins, so certainly this pathway can happen. Okay. We talked yesterday or a couple of days ago about photostability of fluorescent proteins versus organic dyes. And so I happened upon this and I loved it because it just says right here, organic dyes are brighter than fluorescent proteins, says Luke Lavis. Okay, but I also actually found evidence from one of our other speakers. So in this review here, Philip Tinnefeld's, there's a statement that the photostability of fluorescent proteins is typically about 1/10 that of organic dyes. I might have said 1/100th, so I think we have a ballpark range that there seems to be general agreement. What I can't find is direct comparisons. So, but there seems to be a general consensus that fluorescent proteins are less photostable and probably somewhere 1/10 to 1/100th. Okay, and photobleaching is a real problem for live cell imaging. So this just basically shows you what happens. You're trying to image and your signal goes away. So how do you minimize photobleaching? Certainly one way to do it is by minimizing light exposure. So decreasing your exposure time, decreasing your laser power. If you're doing wide field imaging, you can put neutral density filters and just decrease the transmission of your arc lamp to your sample, or changing your acquisition time. We do a lot of long-term imaging, so imaging over 2 to 4 days in my lab, and we have to collect images every 12 to 15 minutes. If we tried to collect images every minute, we wouldn't be able to do it. So that would be an example of decreasing acquisition time. There's sort of a growing push, perhaps, towards adding chemical scavengers. The single molecule field has done this for a very long time, adding reductants and other scavengers to try to lower, for example, the amount of oxygen in a sample. And Thermo Fisher is trying to market, at least, these products. So this is an example of one of those products here, probably from a Thermo Fisher advertisement. We've never used them, so I can't really say out of personal experience how well they work, but this is one approach to try to prolong your imaging, is to use these kinds of scavengers. Okay. So a note on, or you can choose your fluorescent protein judiciously. Choose one that's more photostable. The challenge is that it's rarely possible to compare the rate of photobleaching of different FPs from different papers. Relative measurements within a single publication are far more informative and reliable, that you can see under these conditions, the relative ordering. But the most important thing is really to assess it under your own conditions, because wide field, the photostability under wide field illumination is different from spinning disk, which will be different from laser scanning confocal. And even worse is how you want to use the fluorophore. So in vitro measurements are very different than in bacteria, which is different from yeast, which is different from mammalian cells. And even in mammalian cells, the photobleaching in different organelles will be different because of the different chemical environment. Okay, so how is photobleaching typically reported? This guide from 2008, I think, was the first to really sort of establish a norm for the field. And what they acknowledge is that due to differences in the efficiency of excitation for any given optical setup, the photobleaching curve should be normalized to account for how well you absorb a photon and how well you emit a photon, how well your emission spectrum overlaps with your excitation source, the transmission spectra of the filters, so what are your filters actually filtering out, and then other optical components of the microscope. So the metric that they came up with is that if you start with an emission rate of 1,000 photons per second, that the time to bleach would be to go from 1,000 photons per second to 500 photons per second. So this is the key here, so for each fluorophore that you're looking at, you don't excite them all the same. You actually excite them differently depending on their properties, such that you always start with 1,000 photons per second. And then from there, the question is how much time to decrease that rate by half. And what they suggest in this paper is that the dimmer probes require higher excitation power or longer exposure to get to this rate, but that provides a more realistic measure for how you might use these in microscopy. If your probe is dimmer, you are going to have to excite it with more intense light in order to see it inside of a cell. Okay, and then measurements are done in different ways. So purified protein is often measured in these embedded in polyacrylamide, and this is really nicely, there's an entire protocol in the supplementary information of this paper. Purified protein is also measured in microdroplets and can also be and should also be measured in cells, typically in the nucleus, so that you can illuminate the entire nucleus and you avoid diffusion artifacts. Okay. What I love about this paper, and in fact what I love about much of Roger's writing, is for anybody who knew him, he had a very, he was very witty, and he liked to write papers in a way that really told everybody the truth about things. And so in the supporting information of this paper, I think there's a really nice commentary about photobleaching that often gets lost or forgotten. So he said, "We must admit that our numerical estimates of photobleaching have undergone some systematic revisions in successive publications." And then he goes through and details them so it's more accurate to perform. He's talking about these summations that give rise to these estimates over wavelengths rather than to assume monochromaticity. The mineral oil matters, so you have to carefully prepare the mineral oil. Many fluorescent proteins themselves refuse to bleach with single exponentials, so they have a fast phase. And then, of course, non-uniform illumination causes a lot of problems. So it's really a way of acknowledging the complexity of making these sorts of measurements. Okay, so this is an example of what he's referring to about fluorescent proteins refusing to have a single exponential decay. So M cherry is actually pretty good. This is the fluorescence decay upon illumination. And so here's that 50% mark. So you go from 1,000 photons per second to 500. But you compare that to M emerald and you see this rapid decay followed by actually a very slow phase. So here's the 50% mark for M emerald, and you can see that if you just use that T1 half, you're going to predict that M cherry is really much better than M emerald. However, you see that after that fast phase, this really doesn't photobleach very much at all. And so this particular metric may not necessarily be predictive of your performance in microscopy. Okay, and it actually gets worse because if you look at these fluorescence decay curves under different illumination conditions, lower intensity versus higher intensity, you see wildly different results. So there are single exponential decays, there's multi-exponential decays, there's kindling where it actually increases and then decreases. My favorite is over here. I think you get this rapid decrease followed by kindling, followed by photobleaching. Okay, so it's very complicated. Okay, so what causes this complicated behavior at the ensemble level? All right, we've referred to dark states. So it's related to conversion to dark states. And so I want to talk a little bit about this conversion to dark states and how we now know that that rapid decay is related to conversion to transient dark states. So the first observation of blinking or dark state conversion in a fluorescent protein was by Robert Dixon in W.E. Murner's lab, and these data were actually shown in one of the earlier talks where they noticed at the single molecule level there are fluorescence fluctuations of a single molecule. When they tracked this single molecule over time, I really like the text here. It tells you each GFP molecule generally ended in a non-fluorescent state after about 10 to the 6 photons. But the final dark state was found to be long-lived because after 5 minutes of no pumping, so they gave it a rest, and then they illuminated with 488, and what they found is that this bright state could be recovered. And that's really shown in these panels here, where here it's in the dark state, and then after this period of rest, they illuminate it and they turn it back on. Then it'll go through some blinking, but basically it ends in a dark state. They can turn it back on, so they could do this over time. It's very insightful. In this paper, they proposed this following model, theoretical model for blinking. So if you started in this ground state here, you could get excited. They deduced that the blinking state, that this intermediate state must exist, that I must be a dark state that can spontaneously relax back down to the ground state because of the fact that it blinked. So without doing anything to the sample, you could get fluorescence to come back, which suggested that this was a spontaneous relaxation. However, they also noticed that after a while, the whole fluorescent protein went into a dark state and they had to hit it with light to get it back out. And that they proposed as this state here, N, that upon illumination, now you could kick it out of this dark state. They proposed that this state here they called it N for neutral, so they proposed that the chromophore got protonated at this time, although they didn't have experimental evidence for that. The experimental evidence came later through some very nice physical chemistry studies that really established what was happening is that a proton is transferred in the excited state. So how do you do that? You need a spectroscopic measurement that's sensitive to excited states. So in physical chemistry, these are transient absorption or transient fluorescence processes. So you need very fast time-resolved measurements because the lifetime, the fluorescence lifetime of a fluorescent protein is between 1 and 5 nanoseconds. So your time resolution has to be pretty fast. So this was first established in Steve Boxer's lab and then extended around the same time in another lab where the Boxer lab used this process of fluorescence up conversion, but basically they were doing time-resolved spectroscopy. How do they know it's proton transfer? They did the measurements in H2O and they did them in a deuterated solvent. So D2O, the deuterated solvent, a D is heavier than an H and typically gives rise to a kinetic isotope effect. So if something happens slower in D2O, that suggests that there's a proton involved, an exchangeable proton involved in the process. And that was indeed what they found here. So in 1996, this is very early on. It's not far from it's not far from these demonstrations of blinking. In fact, this happened before the demonstration of blinking. That may be why Myrner proposed that it was excited state proton transfer. So they happened around the same time, but it really was only recently that we had a true atomic picture of what this might look like. So in 2009, there was this beautiful paper on femtosecond stimulated Raman spectroscopy. And there's a lot of details here, but suffice it to say that when you look at the Raman shift, it tells you something about specific chemical signatures. So you could look at, for example, a CH, a particular mode of a CH bond, or a stretching of a CO bond, or of a CN bond. So you can look at very specific bonding signatures. And this study was done, it's femtosecond spectroscopy, so if you see the timing here, it's very time resolved. You have 100 femtoseconds, 200, 500 picoseconds. So it's accessing what are some of the chemical changes at a really fast time scale. And essentially what they deduced from a really detailed study is that, I think I put it here. So when you excite the chromophore, the pKa of that tyrosine gets lowered, so you shift electron density and you make that proton more acidic. Okay, so if it's more acidic, if you remember, an acid wants to donate a proton. And so if you make that proton more acidic, at the same time, the CO actually shifts up towards this water molecule here. And so that proton has become more acidic and it's getting closer to that water molecule. And that facilitates the transfer of the proton from the protonated chromophore through the relay. So what I find this really striking here is it's really these advances in physical chemistry, it's spectroscopy that led to this insight of what is happening here. Okay. So why is dark state conversion? Why is this process of blinking a problem? We saw last night a really beautiful and scary example of why blinking is a problem for palm, for super resolution imaging, and that it can lead to over-clustering. But why is it a problem if you're doing wide field imaging or typical live cell imaging? So the problem is that the dark state conversion diminishes your photon output. So there's this really nice study done in 2007 out of Stefan Hell's lab where they illuminated a sample with pulsed excitation, and they used a laser where they could tune the repetition rate, sort of the timing between pulses. And what they found is that the total fluorescent signal depends on the timing between the pulses. And to me, when this paper came out, it was really transformative. It was very striking because that doesn't work anymore. Okay, so because what they showed is that if you just look at GFP, and what you're looking here is essentially like the fluorescence output as a function of this, the timing between pulses. So this is more time between pulses, less time. So the more time between pulses, you get 20 times the amount of photons out of GFP. That suggests a lot of the protein is going into a dark state, and you're not getting signal from it. But if you could change your illumination scheme such that you allow time for the protein to relax, then you can get more light out. It also suggests that where many of us are doing experiments, we have low signals, not just because the fluorophores aren't as bright as we would want them to be, but because they're converting to a dark state and they're not giving us photons to measure. And so this is just an example that here, if we excite and it's converting to, the molecules are converting to a dark state, if it relaxes on this microsecond time scale, if you allow for relaxation from that dark state before you hit it with light again, then you're going to have a more productive photocycle, you'll get out more photons. They also show that if you continue to excite the molecule, so if we use these fast repetition rates and we continue to excite the molecule while it's in a dark state, then we can ultimately just bleach the molecule. Okay, so this millisecond pulsed illumination can separate this dark state conversion from this photobleaching, and this is an example of that. So this is an example of a pulsed excitation. These are about 2 milliseconds wide, and then you could tune this rep rate, the timing between pulses. What you typically see when you do this is that when you turn on illumination, you see fluorescence and it can rapidly decay. You turn off the light, the signal goes down. You wait and you do this again and you get a pattern like this. So you'll notice that within this one pulse, you see this fluorescence decay within 2 milliseconds. But when you turn on the pulse again, you see that the fluorescence actually goes up. So some of it recovered. What was recovered was reversible. So it suggested that that was dark state conversion. That amount of the fluorescence change was due to dark state conversion. And of course, what remains is what is the actual amount of photobleaching. So pulsed illumination allows differentiation of the photobleaching and the dark state conversion at the ensemble level. Of course, this is trivial at the single molecule level because you can just watch everything. But at the ensemble level, this kind of a scheme allows you to differentiate these. Okay, so we took, a number of years ago, we took on a study to try to understand photobleaching and dark state conversion in a series of related proteins. So we took this family, these 2 red fluorescent proteins only differ by 4 mutations, and so we thought this would be a compact study to examine the influence of individual mutations on photostability and dark state conversion, and then we added in a few other mutations for the RFP aficionados. We expressed them in the nucleus of HeLa cells, so this is what it would look like, and then you bleach and there's no signal. We made a series of measurements, so we continuously illuminated with a xenon arc lamp. This is like wide field imaging, or a CW laser at a variety of intensities. We also looked at pulsed illumination, like we just talked about, so 2 millisecond pulses, and then separated by different periods of dark time. So this is an example of what you might see. This is continuous illumination and pulsed illumination. You generate these patterns where they go up and they go down, but the tips of all of these is really what represents photobleaching under the pulsed illumination conditions. And essentially, making both of these measurements and doing some modeling, what we realized is that this fast phase here is really dark state conversion. So measuring by both methods allowed us to differentiate that the biggest contribution to this fast phase is this conversion to a transient dark state. Okay, so what did we learn? We learned that things are very complicated. So you don't have to read any of this, but I want to walk you through what this is plotting and just point out 2 examples. So not surprisingly, there's a correlation between photostability at different illumination intensities. So this is just comparing lower illumination intensity, higher illumination intensity, and then going that direction. If you're over here, you're more photostable. If you're over here, you're more photostable. So there's a general correlation. So if you look at the black circles, they kind of all fall on a line here. So that if you're more photostable at low power, you're probably more photostable at high power. But there are some distinct outliers that are interesting. So here, if you just look at these two fluorescent proteins, what you see is that comparatively, M cherry here is much more photostable at low power compared to high power. So it's really off of this general trend line. And conversely, this particular mutant is a lot, it's a lot less stable at low power compared to what it is at high power. Okay. We also looked at how readily a protein converts to the dark state compared to its fluorescence lifetime. So the fluorescence lifetime essentially determines how much time the molecule is spending in the excited state. And what we find is that there is a general correlation that you might expect. So as the lifetime increases, generally there's a greater probability that you're going to convert to a dark state. Which is not surprising, but the correlation is actually pretty weak. So what the correlation suggests is that there might be ways to promote or to minimize this correlation. So if you want to promote dark state conversion or prevent dark state conversion, you might be able to do that without manipulating the lifetime too much. Okay. However, the actual connection between dark state conversion and photobleaching itself, which is what we were trying to get at, is really complicated. And that's illustrated. I just want to highlight 3 or 4 points on this plot. So this is a plot of the percent dark state conversion compared to the photostability. So if you're over here, then you're more photostable. So if we just look at one protein, this is a red fluorescent protein. You introduce a single point mutation and you don't change the photostability at all, but you can take the dark state conversion very low, so up from about 70% to about 25%. So they're not always connected. You can take that same protein and make one single point mutation, and we don't change the dark state conversion at all, but we can dramatically increase the photostability. But there are other times where we make one single point mutation and we do both. So what this cycle suggests is that in this case, the dark state is very reactive. So when you're in the dark state, you are likely bleaching the molecule from the dark state. When you're in this state, the dark state may actually be photoprotective. So you're probably not bleaching from the dark state. And what it suggests is that even with a single mutation from one protein to another, we probably are changing the mechanism of photobleaching and dark state conversion, and that there are different mechanistic pathways in different proteins. Okay, so I teach introductory chemistry to undergraduate students, and when I do, they have to learn quantum mechanics. And every time they learn quantum mechanics, they're kind of beating their heads against the table that it's so complicated. So in order to make them feel better, I share these quotes with them from the founding fathers of quantum mechanics. So Schrödinger said, "I don't like it. I'm sorry I ever had anything to do with it." Niels Bohr notes that, if someone is not shocked when they first come across quantum theory, then they must not have understood it. And this is my favorite. I can safely say that nobody understands quantum mechanics, from Richard Feynman. So to some extent, fluorescent protein photophysics is a little bit like this. It's pretty confounding, and we could maybe even replace quantum mechanics with nobody understands fluorescent protein photophysics. At least when we started these measurements, I think we thought we would reveal this wonderful insight just by measuring closely related proteins. But that was not the case, and what needed to happen was people like Dominique's lab and work from other people where people are trying to figure out what these dark states really are. Okay, so I wanted to just highlight two examples where of studies using either time-resolved spectroscopy or time-resolved crystallography in order to try to get at what is the nature of these dark states, because then maybe we can better understand them and use that information for engineering. Okay, so this is the work of Virgil and Ulrich and Dominique, where they were looking at this iris FP that you've heard about. And what they found, probably serendipitously, you can tell me if that's true, but what they found is when they looked at a crystal, okay, and exposed it to X-rays, they found that even at cryo temperatures, the crystals showed a rapid fluorescence decay in the X-ray beam. But you could turn the X-ray beam off and you could get some recovery. And then you could warm the crystal to room temperature and get this further recovery. And then you could repeat the cycle. Okay, so whatever is happening, it's not destructive. Was that serendipitous, or did you try to find this? Okay. 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 We don't know that. Yes. Okay. So what this suggested is that you could then use crystallography to try to access what are some of these states that one sees even in the crystal. And so to make a long story short, they solved the structure and they showed these shifts in electron density. So you can look at what are some of the changes that are actually happening in some of these dark states that are forming. And one of the main things they saw is this distortion of the methylene bridge, which suggests that you're losing pi conjugation. Okay, so if you're losing pi conjugation, then it's not going to be fluorescent. So that can explain why it was dark. And I think they coupled these studies with simulations to show that this could be generated by electron transfer to a triplet state. So now this happened in an X-ray beam. So X-rays are known to induce electron redox changes in samples. But what I think is really powerful is what they suggest is the state wasn't accessed the same way that maybe it was accessed upon a regular photocycle where you excite the molecule and maybe it goes through a series of transformations. So it's possible that the X-rays are inducing a change that's accessing the same sites but in a different way. And so this suggests that we can study these states and gain some insight into what's happening in these dark states. I don't know, it's a little hard to talk about somebody else's work when they're in the audience. Okay, that's what I got out of this paper. Okay, this is another really beautiful example. This is a very recent example of information on excited state structures from time-resolved spectroscopy and crystallography. It's from this group. There were so many authors, I couldn't put them all on here. They were looking at RSGFP, or I guess RSGFP2. And first they did time-resolved spectroscopy. So they were looking at the change in bands, basically at very fast timescales. So you see this rapidly goes away, this rapidly goes away, and this is all done in solution. And then they can fit this to get a kinetic model. So they can look at what forms and what goes away. And essentially what they see are this one very fast species that comes up, I guess, in 90 femtoseconds, another one that decays in 0.9 picoseconds, and a third one that decays in 3.6 picoseconds. Okay, so there's these three species. They suggest this is formation of the excited state, and this is this is formation of two excited states, and this is decay of those two states. Then what they did is this time-resolved femtosecond crystallography. I had never heard of this. I'm not a structural biologist. I had never heard of this until this paper. And it was, I think this is the first report of this general method. The idea is that you have a stream of small crystals, like microcrystals or nanocrystals, that you shoot through the X-ray beam. and you rapidly collect a diffraction pattern. So this is really, really, really fast. And so how do you build a structure out of this? You do it by basically summing or using the diffraction pattern from thousands of individual crystals. So this is a way then of being able to get structural snapshots, but at a very fast time scale. So in this paper, they use, coupled with this transient absorption or time-resolved absorption, they use this technique, and basically I'm skipping like the whole paper in 30 supplemental figures to show you what they discovered, which is that they found a twisted intermediate. So they found a new intermediate in this RSGFP2 that they called a twisted intermediate. So the off state is in gray and the twisted intermediate is in red. And what you can see is there's a twist of the chromophore that ultimately leads to this downward shift of this central helix that holds the chromophore. Okay, so why is this important? Well, what I loved about this paper is they showed that, this twisted intermediate revealed that there is this one residue here, this valine 151, that in the twisted chromophore would have a steric clash with the chromophore itself. So I'm trying to show that here where you see the twist and you see this shift of this valine 151. So they then use this to suggest maybe if you make this valine 151 a little bit smaller, you make the residue at that position a little bit smaller, that you could actually facilitate the photo-switching of the protein. So they made a single point mutation of valine to alanine and showed that the switching kinetics, I guess, don't change that much. The quantum yield of switching increases. It almost doubles. So to me, what this suggested is that you can use spectroscopy and crystallography to gain insight into the time scale of the processes, the nature of those processes, and maybe what some of those structures are. And that with that information, it might provide insight into how to change the properties of the proteins that people are trying to use. So this is your take home if you were asleep for the last hour. Photophysics are complicated and not well understood. Different FPs have different properties. Understanding the nature of the excited state transitions and the structure of these dark states can provide insight into how to tune and engineer these properties. And so these types of techniques, I think, are really powerful to provide some of that insight. Okay. But we also, so this is like how you could make the probes different or better, maybe. But we also can take these bugs and turn them into features. So no property is all bad. It really depends on your perspective. So how have some of these properties, such as dark state conversion, been leveraged for microscopy? So even photobleaching has its perks. How many people know of FRAP? Fluorescence. Okay, so everybody knows about it. So if you frap, you have to photobleach, and you actually don't want your protein to be that photostable because you want to be able to frap it really quickly and then watch recovery. So frap is this process where you photobleach a region and then you measure diffusion. It's kind of what people did before photoactivation, I guess. So here's an example of frap just showing if you photobleach 2 spots here, you can see recovery is much faster in one compared to another. This is a snapshot of those particular data, and then something like this can be used to show that one recovers more slowly, and that in this case it's a transcription factor and it recovers more slowly when it doesn't diffuse and it's bound to DNA. So you can look at mobile and immobile fractions with photobleaching. So photobleaching isn't always bad because when you do palm, you have to, the successive activations, you have to have your fluorophore bleach before you can do the next round. So if you were doing live cell palm, then you want your bleaching to be, relatively fast. You want to collect as much photons as you can, but then you want it to be relatively fast so you can collect rapid acquisition. But generally, photobleaching is a problem that we need to fix. Okay, but what about blinking? So there are We've already had some very nice talks on photochromism and the exploitation of photochromism for localization microscopies. But there are also blinking microscopies, I guess you could call them, or microscopies that leverage the fact that there's bright and dark states, transient or spontaneous transitions between bright and dark states, or the fact that if you have probes that have differences in photo switching or in dark states, that you could lock in on signals from different kinds of probes. So Peter briefly introduced this, so I probably don't have to cover it in too much detail, but I will just mention it because he had to rush through his blinking sensors, and I wanted to highlight that. So use of blinking, I'm not positive, I think this is the first report of SOFI, which is super resolution optical fluctuation imaging. And as the name suggests, it's fast, it's background free, and you can use it with a simple microscope system, which I think is one of its advantages. So it relies on a statistical analysis of these stochastic fluctuations or blinking. It can give you, at least in this paper, I think about a 5-fold improvement in optical resolution with a typical wide field microscope and a CCD camera. So what I like is no special fluorophore, no special microscope, leverages dark state conversion, but it does require some sophisticated analysis that Peter is an expert in. 3 conditions have to be met. So the fluorophore needs to exist in 2 different states that are optically distinguishable. Individual emitters need to switch independently and over. And then images should be acquired with pixels that are smaller than diffraction limit. So this is an example of like the SOFI pipeline here. So you basically collect images over time, and then you have in each individual pixel, you have fluctuations, and then the fluctuation in each pixel is really going to be the sum of the fluctuations from all of the emitters that overlap that pixel. So then you do some nice post-acquisition analysis that allows you to look at correlation functions and then isolate the individual emitters. This is a picture from that original paper. But what Peter did, actually, when he was a postdoc with Jin, was to take this and apply it, use fluorescent proteins, and to take this and apply it to live cells with something called photochromic stochastic optical fluctuation imaging, or PC-SOFI. So again, this analyzes these temporal fluctuations. It's easy to implement and compatible with high-speed data acquisition, so this is good for live cell imaging. And these are just 2 examples of images that they collected. Okay, so this allows you to get better resolution on live cells very quickly, but I think one of the coolest examples, potentially the coolest example of using blinking is what Peter talked about at the very end of his talk, which are biosensors that actually exploit optical fluctuations. Okay, so this is the general principle. Okay, so I should back up and say, if you look at TAG-T, so this is a fluorescent protein, you look at single molecules, you look at their optical fluctuations, and it's kind of like that. If you take that same protein and you fuse it to something else, in this case Drompa, and you look at the fluctuations, you see much greater fluctuations. And so they termed this fluorescence fluctuation increase by contact, or FLINC. And so the idea is that something about bringing Drompa close to TAG-T is changing the blinking properties of TAG-T. Okay, such that you're now increasing the fluctuations when they're in close contact. So it's not hard to imagine, how does this happen? They postulate that there's an interaction between Drompa and this other fluorescent protein, such that there's a basic pocket and an acidic patch here, and those both seem to be important for this proximity-induced increase in blinking. And you can actually see a difference in this dark state conversion. You can see here that there's a greater fraction of molecules that convert to the dark state when this tag T is fused to Drampa. Okay, so it's not hard to imagine how you could turn this into a sensor since it depends on proximity. And they used the pKa sensor scaffold. So this is a sensor for protein kinase A. So they took off the regular boring CFP, YFP fluorescent proteins, put on Drampa and tag T, and now you have a sensor where the readout is a change in optical fluctuations of the tag T. Okay, and so they did, Peter already introduced this, pKa is really important. Cyclic AMP was the first second messenger ever discovered back in like 1972-ish was the Nobel Prize. But the challenge is that there are a ton of things that produce cyclic AMP in cells, and pKa helps to decode that cyclic AMP signal. So it's a really important signaling molecule, signal transducer. And in making a sensor, what they found is that if you measure the fluctuations before you activate the kinase, you would see something like this. When you then stimulate the kinase, you see an increase in signal. That's an increase in these fluctuations, which suggests an increase in pKa. And you can inhibit this with an inhibitor. And so this is now looking at the activity distribution of an enzyme with super resolution. Rather than just where the molecules are, it's the activity of that kinase. And I think in one of the prettiest images in the paper, they looked at the activity gradient at super resolution in a migrating cell. So you can see it's not homogeneous. You have a buildup at this edge here, and you can see in particular this increase in activity in these protrusions. Okay. I kind of want to, should I stop and give you guys time for questions? Yeah, but you want some questions? Maybe I'll talk a little bit about microfluidics and how you can use it to measure photophysics, just to give you a sense of the kinds of things that we're trying to do. But I'll try to do it in just like 5 minutes so you can have plenty of time for questions. So in my lab, we develop fluorescent tools to study live cells. Because we feel the best probes are often those motivated by biology, we're actually very much, and in fact most of our work is using model systems to try to understand how cells manage metal ions or inorganic ions and RNA, and then studying host pathogen interactions. But in doing this, we need to develop better probes. We need brighter fluorophores, we need more photostable fluorophores, and we might want to manipulate the properties such as blinking and some of these photophysical properties. So we started developing these microfluidic sorters so that we could measure properties at the ensemble level in high throughput. So these were some of our questions. Can we explicitly engineer properties like photostability, dark state conversion, fluorescence lifetime, and carry out multi-parameter screens so that we don't optimize one property at the expense of another? So how can we improve photostability? If you look at our favorite photocycle here and you look at your typical equations for quantum yield, lifetime, shown here, and I'll just remind you that this K and R is like many, many things wrapped in, dark state conversion, et cetera, et cetera. So first you might look at this diagram and say maybe we should just decrease the lifetime, because remember, long lifetime can give you more dark state conversion. But the problem is if you look at a correlation plot of lifetime and quantum yield, they're like directly correlated. So if you decrease the lifetime, you're probably going to decrease your quantum yield. So that probably won't work. Some other options are, could you explicitly decrease the lifetime of a triplet state, decrease excited state absorption if you could turn off some of these processes, decrease other excited state reaction pathways. So if oxygen accessibility to the chromophore is a problem, maybe you can block oxygen access pathways. Maybe you could actually drive this to convert to a photoprotective dark state if you knew what that would look like, or really try to increase this radiative rate at the expense of these other properties. So we started developing a microfluidic device. This is obviously not microfluidic. The microfluidic is there. It's just an optically transparent small device that you can couple optical measurements into. The advantage of microfluidics is you have precise flow and environmental control. We can monitor single cells. It uses small liquid volumes. It's compatible with lots of measurements, and we can actually select cells with properties. So this is the concept here. If you have cells with a fluorescent probe, they're flowing, and you can go through a series of interrogation spots. So it's conceptually similar to FACS, but you can make multiple time-resolved measurements on the same cell before you sort it. So we've gone through many versions of this kind of a microfluidic. We started with this kind of a geometry where we measure fluorescence. We use a really intense beam to try to bleach the protein, and then we measure fluorescence again. And ideally, we would just select proteins that don't bleach much. The challenge is that the cell is only in this beam for about 2 milliseconds, and it's very hard to bleach without using a really, really high laser intensity. You can do it, but then that's not terribly relevant for imaging where people aren't using those powers. So we then switched to a model where we have the cells go through many beams to simulate pulsed excitation. And indeed, this geometry allows us to identify and select for dark state conversion, if that's something we want. When we implemented this, we actually took this first beam and we used a modulated, I did it again, sorry. We modulated this, so we use frequency. It's modulated at a certain frequency such that the emission is shifted from excitation, and that phase shift allows you to measure the fluorescence lifetime. So in this geometry, we measure the lifetime, and then we measure dark state conversion. We then implemented a simpler mode where we just measured lifetime and brightness, and then sorted cells. So we have a bunch of these different kinds of geometries. And this is the premise of how you might use this to select properties. You can generate a library of proteins, and you can put them into cells by viral transduction. So each cell only expresses one library member. And then you can make these measurements on cells. You can sort them, and then you can take them through multiple rounds. And this is an example of how you might do that. So here each of these dots is an individual cell expressing a particular fluorescent protein variant. So we made a library of them. And we measure the lifetime, and this is a parameter proportional to fluorescence, so the fluorescence intensity. So we measure lifetime and intensity. And this is a library here. If you select this population with a long lifetime, you can make mutants where you shift the lifetime and the brightness of a fluorescent protein. So M cherry has a lifetime of about 1.8 nanoseconds. And here we shifted it up to a lifetime of about 3.5 nanoseconds. So this is just an example of how you can take this kind of technology and now in a high throughput way start to make measurements, and in a high throughput way start to tune the fluorescent protein scaffold such that you can select out proteins that would have different properties. Okay, so I am going to end there, but I want to show you, I'm just going to skip all this. We mostly do live cell imaging. So I'm a little insecure talking about all of this in front of some of these experts. But okay, so this is a picture of my lab. I actually cut out our mountains. So Boulder is right at the base of the Rocky Mountains in Colorado. So there's beautiful Flatirons and the Rocky Mountains right behind us. But this is a picture of my lab from last summer. These guys are all postdocs, grad students, and then I usually have 3 to 4 undergraduate researchers in my lab. Our work, the only work of ours I really talked about was work looking at some of these photophysics and microfluidics, and we have done that with a close collaboration with Ralph Jimenez and Jilla. So thank you for your attention, and I'm happy to try to answer your questions.
Thank you, Amy. Questions?
Thanks for the talk. I have a question on this flow cell.
Yeah.
Well, I don't know if you could call it at the chamber. So how exactly, what kind of properties do you measure and how do you do it? I didn't understand.
Okay. Yeah, sorry. It always happens when you try to
It
go
measures
fast at
like
the end.
different parts of the journey of the cell.
Yeah, so you make, I'll see if I can, I'll take it to here because I think that this might make it easiest. So here's an example. If the cell is flowing, each of these green squares is supposed to represent like an excitation beam. So the cell goes through there and you excite the fluorescent protein and then you measure something. So in the first beam, we measure the fluorescent, in all beams, we're actually measuring the fluorescent signals. We're measuring fluorescence emission. And in the first one, we can get information because we're using this modulated light here. We're not just measuring the intensity, we're measuring the phase shift between between this pattern of the excitation light and between that pattern of the emission light. So it gets phase shifted. It's phase shifted because of the fluorescent, the time the molecule spends in the excited state, which is the fluorescence lifetime. So in this particular geometry here, we can measure what is the fluorescence lifetime of that protein in that cell. And then in all of these other beams, we're just measuring the total fluorescence emission. So if the molecule is photobleaching, then you see something like this. The fluorescence intensity goes down over time. And so we write a program that collects that information, what is the lifetime and what is the fluorescence intensity at each of these spots, very quickly. So it can then say, do I like these properties or do I not like them? And if I like them, then I sort the cell. So I apply, in this case, an optical trap, and I keep it, or I don't, and I throw it away. And so that allows us to just make measurements of fluorescence lifetime and say dark state conversion on lots and lots and lots of different mutants, so that we can see how diverse the landscape is. If we want to make the properties better, say we want to make the lifetime longer, that's where we would take this sort of approach. What the pink represents is we set that a gate so that if you have a lifetime up here, we want to keep you, and then it'll sort that cell. And then we can collect those, and if we run them through this device again, we see, okay, indeed the population has shifted. So now we have cells that have fluorescent proteins, and all those fluorescent proteins have long lifetimes.
And do you think you could use that to improve other properties of the proteins, not just photobleaching?
Yeah, so the geometry, so the reason I show all of these geometries is to show you that it's really adaptable for measuring a lot of different things. Particularly, so the challenge is our time window, because the cells are flowing, our time window is about 10 seconds total. So that means, so although through all of this we were trying to ultimately engineer photostability, we need more time for photostability. So we now have a different geometry where the cells kind of take a meander through a long walk and spend a lot of time under illumination in order to try to get at the kind of photobleaching we experience when we do microscopy. You can bleach with a really intense laser, but nobody uses that for microscopy. So if we want to make better proteins for imaging, then we have to mimic selections under imaging conditions. But you could, for example, interleave 405, 488, 405, 488. Or so you could try to look at switching kinds of behaviors.
More questions?
Peter. Did I did
No,
did I butcher Flink or Sophie? I
no,
hope
not at all.
Okay.
That was fine. Yeah, thanks for the great talk. But I'm just like intrigued, this 2005 paper by the Hell Group with
Ye-
this relaxation.
Yeah.
I saw that and I was like, hey, this is really nice. And
Yeah.
then I never really tried it. But have you played with this?
Oh, using pulsed
Like,
excitation?
right, like exciting for a microsecond and then waiting a microsecond.
Yeah,
or-
yeah. I mean, so it works. It happens. We didn't adapt. I mean, we do a lot of live cell imaging in a core facility, so we don't have a lot of flexibility. We haven't adapted a microscope and then done live cell imaging under those conditions because we don't have the flexibility to adapt our microscopes in that way. But if we built a rig that allowed us to do this pulsed excitation, I don't think we re-quantified GFP, but when we looked at RFPs, which is mostly what we've looked at, yeah, you see if you pulse, you can get out a lot more light out of the red FPs. It really just depends on how much dark state. So we haven't tried imaging that way yet, but I think it's true that the pulsed excitation gives you, on the whole, more photons.
Thanks.
If not, I have a small question.
Okay.
How in your lab do you avoid the problem of photo bleaching? Okay, you
Yeah.
use less acquisition time, but for days, do you simply change the protein and or the fluorophore, or do you actually?
Yeah, so we spend a lot of time trying to do like long-term imaging. So I'll show you just one movie of something that we're doing. So we study the mammalian cell cycle, so cells dividing. So this is a movie, a zoom in on cells that are expressing an M cherry attached to H2B, which makes it attached essentially to DNA in the nucleus. And what's nice about that is you can then use it to track cells during mitosis. So then we use that to image cells over long periods of time. So each of these spots is a nucleus, and this movie is over like 48 hours so that we can watch cells as they divide. And it's a little hard to see, but when a cell divides, the code changes number and changes color so we can identify these division events. So why do we want to do this over 48 hours? We want to study the cell cycle, but really we want to study individual ions and molecules that are involved in the cell cycle. So we make a lot of biosensors, and we're monitoring, for example, zinc over the course of the cell cycle. So what are our challenges? So one challenge is here, we use M-cherry. M-cherry, like for all of its problems, actually is pretty darn good for long-term live cell imaging. These images are collected every 12 minutes. If we try collecting more frequently, like every 5 minutes, then we lose our signal too quickly. So what do we do in that case? We've tried a bunch of chemical manipulation of the media, but for 48 hours, that's a real problem. You change the cell biology. So we can't use scavengers. So in this case, we don't have a brighter, more photostable protein, so we just image less frequently in order to fix that problem. But it's one of the reasons why we want proteins that are brighter and more photostable, so that we can carry out these kind of measurements for longer. Also, this is not CRISPR'd into the genome. This is exogenous, and that is a problem for some of our measurements. So we're doing a lot of single fluorescent protein reporters, some of these kinase translocation reporters, and we find that the signal intensity is a real problem with those as well. So in general, the fluorescent proteins need to be brighter so that you can put them into the genome at endogenous levels so you can perturb the biology to a minimal extent. I don't know if that answers your question.
Okay. Quick question. Okay.
Yeah. Yeah, no, that's okay. So this is a very good question. So we actually can do both. So we can do bacteria, we can do yeast, and we can do mammalian cells. And we have used all three. So for fluorescent protein engineering, there are some advantages of using bacteria, one of which is that the maturation in bacteria is kind of reminiscent of maturation in mammalian cells. There's not always a one-to-one correspondence, but it's a reasonable predictor. If something matures well in bacteria, probably will mature well in mammalian cells. In bacteria, you can use large libraries, which is great. We've tried yeast because yeast are similar to bacteria. You can transform them, you can get plasmids out, you can do very large libraries in yeast, but it is eukaryotic. The problem with yeast, I don't know if anyone has ever worked with yeast, but they do not regulate their metabolism very well, and so they get wild pH changes depending on like how much sugar they're exposed to and how dense they are. So yeast can be hard to screen in, we have found the hard way. Mammalian cells we like because mammalian cells are ultimately where we're going to use the fluorescent proteins, and there are certainly notorious examples where something works great in bacteria and it just doesn't fold in mammalian cells, or it's not very bright. So mammalian cells are ideal, the challenge being it's harder to get big libraries in, and then you can't transform them, so getting your library in is a slower process using virus. And then getting your sequence out, you have to usually do high throughput sequencing to get it out because you can't get a plasmid out of a mammalian cell. But good question.
If you still have more questions, you can catch her in the break or in the evening. Once again, thanks a lot, Amy, for the wonderful talk.
Thank you. Sorry, please stay seated for 5 more minutes. I have a couple of announcements to make, plus one slide to actually show you. So just be a little bit patient. The first thing is you will find in your boxes some certificates in case that's useful when you go back to your labs. So certificates that you have attended the school. The second thing is the administrative staff here gave us these papers for you to fill, like what do you think about the school in the sense of the premises. We will also actually ask to fill up an online Google form which will be more connected to the scientific aspect. So please take the time to fill both this and the thing that probably Manu will explain in more details concerning the Google form. The third thing is for those of you who will be leaving before lunch tomorrow, you can ask for lunch boxes. So if you want a lunch box, you just put your name down at the restaurant before tonight. Okay? And then just 2 minutes, please. I'd like to show you actually how we could organize this workshop financially. So usually when you go to a meeting, at some point you will see the sponsors. So you can look at them, but I would like to quickly actually explain you what it is about, because it can be somewhat instructive maybe to see what's the different motivations for all these sponsors to give us some money. So I'll start, if I can. So for you to know, so this Les Ouches school is actually a part, essentially, of the Grenoble University, the UGA. So it's kind of a little satellite. And so it's basically thanks to the Grenoble University that this school exists. And this is strongly made in association with the CNRS, so which is the Centre National de la Recherche Scientifique, the Institut National Polytechnique de Grenoble, which is kind of an engineer school, and also in association with the CEA, which is Commissariat à l'énergie atomique, and also the École nationale supérieure de Lyon. So all these institutes actually support the school, generally speaking. Now, on top of that, so we have obtained some fundings. So it's actually like microcredit kind of thing. You get 1,000 euro here and 2,000 there and 500 other euro elsewhere. So it's a bit of a pain, really. But so you have to sort of adapt every application to the kind of interest that all these lab and institution may have in mind. So one big source of funding was the French-German university, and that was really thanks to the fact that this school is organized with Ulrike, who is from Germany, and so the fact that this is in fact a French-German school made it possible to obtain quite a lot of money from this. It's probably like a 30-page thick application, however. So yeah, we have, like in France, we have this France bioimaging thing and Frisbee thing, which are kind of national infrastructures. And these infrastructures, like France bioimaging, like is around imaging. So they are interested in markers in the first place, that's for sure. Frisbee is more like structural biology kind of stuff. So here we have to sell the school like this is good for cell imaging. Here we have to say like this is good for structural biology. Yeah, we have like GRAL is actually a Grenoble type of excellence funding stuff. So it's also related to structural biology, and since essentially I belong to the one of the lab associated to GRAL, we could get money from that one. Thanks to Manu, essentially, we got good funding from the European Biophysical Society. So that was very useful. And also thanks to Manu, who is part of this so-called GDRI, International Group de Recherche, which associate, I think, a number of French and German laboratories. We could also fund a number of speakers. Now we have these two, like Sociétés Savantes in France, the Société Française de Biophysique and the Société Française of Microscopy. So actually, for example, when we asked money from the Société Française of Microscopy, who is very much into electron microscopy, then it struck our mind that we should have really something about correlative microscopy. So they actually help us to make the program better in a way, because they provide some feedback, and I think it was excellent to have like Rainer in this context. The SFB, so Manu holds the wallet of SFB, and this is really great because we can sort of do a little bit of some fully legal arrangements. Which makes it like a little bit easier to pay for like ski tickets. Yeah, but I think there's no problem with that. It's fully legal. It's no problem. Okay, you will see also la région Auvergne-Rhône-Alpes. So that was kind of strange because they basically have no interest in fluorescent proteins or organic dyes. But when we tell them that we will invite like very smart speakers coming from the US and Europe, then they ask, okay, if they maybe go to Chamonix and look around and find it beautiful, they will maybe come back and spend money in our country, in our region. And so it's kind of a way to get the money is to tell them we will take these people to Aiguille du Midi. Kind of fun. And then to finish with, we have had a number of private companies kindly contributing some little amount, but it's always well received. And Imabio, very importantly. Ooh la la, and Laurent is in the room. So Imabio is another GDR, Groupement de Recherche, which is essentially entirely focused on microscopy of living systems, mostly fluorescence microscopy. And Imabio is in particular in charge of organizing this huge Mifobio fair, which takes place every 2 years, and the next one will be in October. This is a fantastic kind of a school, 1 week long with many, many events. I really recommend if you can join that. And in a way, this present school is kind of can be seen as a kind of a satellite to that, in a sense that we cover like a small niche within this whole field, which is the markers field. Okay, then I think that's it. I didn't forget any of the sponsors. So that was just to tell you how it was made possible. Thanks.
Okay, one more second, please.